I have now the right, nice, red cursor. So I apologize, everyone, and I'll start all over again. Not all over, but so I will bring you to the next slide. So we're interested in dilated cardiomyopathy. It's a disease which has a major contribution from genetic mutations, supposed to be involved in 30 to 35% of the DCM manifestation in clinics in form of sporadic or familial mutations. And it involves many genes involved in very different functions, going from sarcomere, cytoskeleton, cell adhesion, chaperone, splicing, signaling. It's a kind of amazing, the vast diversity of genes which are involved in this disease, which ultimately end up in similar pathology in a way. It's also, of course, a manifestation in mitochondrial disease, and it can also be caused by non-genetic causes such as drugs, toxins, myocarditis. But in many of these cases, actually, genetic predisposition are suspected. So despite this complex and very diverse etiology, there is kind of convergence of pathogenic mechanisms in this disease, where finally you always observe a form of weakening of cardiac muscle contractility. Alteration in energy functions, it's very well characterized in this form of heart disease. And there is then this increase in left ventricular filling in diastole and reduced emptying in systole, which is typically a disease leading to heart failure with reduced ejection fraction in that sense. And some other common pathogenic mechanisms include cardiomyocyte stasis, and some of the DCM also present an increased risk of arrhythmia in the patients. And the risk of heart failure is present in most patients and tend to rise with age and male sex, a bit like normal heart failure. So the cardiomyopathies and myocarditis altogether account for about 1% of deaths caused in Western countries, and it's a major cause of cardiac transplantation in Western countries. So today I will talk about this cellular metabolism and signaling, and then I will tell you how these NAD biosynthetic pathways are altered. There are changes occurring in these pathways in the context of dilated cardiomyopathy. And then I will more specifically develop a topic on some of our most recent and unpublished results, actually, on the role of the nicotinamide riboside kinase, NMRK2, in the heart, which is one of the components of these biosynthetic pathways. So what happened with NAD metabolism? Well, it's been known for several years now by many studies in different fields that there is a perturbation in NAD metabolism associated with aging and with disease, notably chronic disease, degenerative disease. And this NAD, of course, has a very well-known function as a redox coenzyme. You have seen, here the oxidized version of the molecule, which is not in NAD+, and then it's a major coenzyme in fuel oxidation for most energetic compounds like glucose, fatty acids, and ketone bodies, notably. And upon oxidation of these energy substrates, NAD+ gets reduced, and then it's the major electron donor to notably in the mitochondria to the complex one of the electron transport chain, allowing the binding of the proton gradient and the synthesis of ATP by the ATP synthase. So the very core basic mechanism of oxidative phosphorylation relies a lot on this use of this NAD plus to NADH conversion by redox reduction. So, but on top of that, there is an emerging role of NAD in the last 2 decades, where it's been involved in other signaling pathways that I'm going to detail in a minute. It's been shown that in a number of different disease contexts or metabolic syndromes, there is alterations not only in the redox state of NAD plus NADH, which is, of course, occurring in heart failure. And actually, we heard about that recently by Christoph Mark in one of these webinar series, nicely illustrating how it was affected. And the work of Rongqian has shown also how this can be affected in various contexts of heart failure. But there is also observations where it's actually the total pool of NAD which is reduced in a number of contexts, disease contexts, and notably in heart disease and in DCM, as I'm going to show you. So why is that? It's because NAD is used in a very different way by some cells, by some enzymes which are not redox enzymes, which are more signaling molecules, signaling enzymes, which are using NAD as a substrate rather than a coenzyme. And this is the case, and the most famous of them are probably the sirtuins. And as you know, there are 7 of them, but I'm showing mostly 3 of them, which are very important and well described. There is this SIRT1, which is predominantly nuclear, SIRT2 in the cytosol, and SIRT3 in the mitochondria. And these enzymes are deacetylases, which remove acetyl groups which are added to the lysine of some proteins, cellular proteins in the different compartments. And as this acetyl is coming from the acetyl-CoA production by the mitochondrial metabolism. And these sirtuins, the acetylases, they remove this acetyl group, and for doing that, they actually cleave the NAD plus molecule at the level of this N-glycosidic bond, which is highlighted here, releasing the nicotinamide or NAM moiety, and using this part, the ADP ribose, and as an acceptor in that case of acetyl groups. And all these enzymes, and especially SIRT3 and SIRT1, have been shown to be prone to stimulate oxidative metabolism, and for SIRT1 to stimulate notably through PGC1 alpha, but there are other targets of SIRT1 in the nucleus, the transcription of metabolic genes and the process of mitochondrial biogenesis. So in addition of these sirtuins, there are other enzymes which consume NAD, and there is one which has been described recently, which is called SARM1, which is involved more in cell death and apoptosis, but it's mostly shown to play a role so far in neurons and immune cells, and not much studies have shown function in cardiac cells so far. Another major consumer of NAD is this enzyme called PARP1. It's the poly-ADP ribose polymerase 1. And it's an enzyme which is activated upon DNA lesions triggered by ROS. Upon this stimulation, its activation signal, it starts to cleave a vast number of NAD molecules to build a kind of poly-ADP ribose polymer, which is used to actually, as a backbone, some kind of DNA-like backbone to recruit the repair complex and repair these lesions. So it's an important physiological enzyme to respond to ROS alteration and attacks on macromolecules like DNA. However, in some instances, if it's overactivated, it can be bad because it can lead to depletion of NAD cellular pools and actually trigger cell death without possibility to produce ATP, so more like necrosis. And it's actually an enzyme which is inhibited in apoptosis, which requires ATP. And finally, there is one last enzyme which is called CD38, which is an ecto enzyme at the cell membrane, which has been shown to use probably some extracellular pools of NAD+, which can occur in some situations. But more recently, it's been shown to be able also to use some of the intracellular pools of NAD and also of NAD+. It generates ADPR, and a percentage of it generates a cyclic ADPR, which has been shown to be a second messenger triggering the release of calcium by the internal stores at the level of the sarcoplasmic reticulum through the rear channel. And with the NADP substrate generating the nicotinic acid ADP product, it's been shown to stimulate the release of calcium from the lysosomal store. So it's an enzyme which is a major consumer of NAD, and its function has been shown in immunity, in cancer, and there are actually antibodies targeting this CD38. But in cardiac cells, there has been some studies who show that it's involved in the generation of calcium sparks and waves, and maybe could have a pro-arrhythmic role. It's not very well known so far. And basically, all these enzymes cleave this nicotinamide at the level of this N-glycosidic bond. There are some additional enzymes which eventually cleave here between the two phosphates, which are involved in other interesting functions, such as maybe controlling the peroxisome NAD level or removing NAD caps on mRNA. This is one of the last functions proposed for NAD, which is quite interesting, but nothing is known on this side in heart failure. But CD38, PARP1, sirtuins definitely are involved in the process of responding to cardiac stress in heart disease. And so all of them consume NAD+. And again, while the involvement of NAD as a coenzyme in energy metabolism doesn't affect the total pool size of NAD, so the sum of NAD plus and NADH, those signaling pathway, because they really cleave, hydrolyze the molecules, they actually lead to a destruction of NAD+. And so that's obviously compensated by pathways which are called the NAD salvage pathways. And these NAD salvage pathways, they are fed first by dietary precursors and vitamins B3, notably the niacin is a major precursor for NAD, and there are also different forms of vitamins B3, I will get back to that, and eventually also the tryptophan amino acid. So it's a relatively poor precursor of NAD in the sense of you need 10 times more tryptophan than vitamin B3 to yield the same production of NAD. But still, it's an important component of NAD metabolism, for instance, in liver. But beyond these dietary precursors, there is a very important pathway, which is in mammals, especially where these NAD signaling pathways are very active. They cleave the NAD, and when they release this nicotinamide, there is this enzyme called NAMPT, which is involved in recycling this nicotinamide base to regenerate NAD+. It's a very important enzyme for the maintenance of NAD in most mammalian cell types. And actually, we heard about it, so I put a little advertisement here. We heard about it just a month or two ago by Tim Longo and Joseph Borlab, a very interesting talk showing how NAMPT is very crucial to maintain NAD levels in the heart. And I invite you to look at the YouTube registration talk again if you missed that. So altogether, this activity of signaling pathways and processes say that induce a turnover of NAD, which is quite high. And in the heart, it's been calculated by a study by the Rabinovich lab to be around 2.65 hours. So quite high turnover of NAD in the cardiac cells. So let me go now to the second part of my talk and tell you more specifically what happens with the NAD pathways changes in DCM. The way we addressed that question was we studied a deleted cardiomyopathy model in the mice, which is triggered by the deletion of a transcription factor called SRF, for the serum response factor. And it's a very important transcription factor whose activity is regulated by a number of neurohormonal signals and mechanical signals to modulate the activity of this transcription factor, whose targets are many genes involved in cardiac contractility, including first actin, myosin heavy chain, tropomyosin, but also genes involved in sodium calcium exchange, for instance, such as NSX1, or energy transfer, such as the creatine kinase muscle white form. These genes are heavily dependent on the SRF presence to be transcribed and expressed in the heart. And there is also a number of microRNAs, such as MIR1, MIR143, which are under the control of SRF. So this SRF transcription factor has been shown to have an activity which is modulated in the context of cardiac remodeling and heart disease. And notably, to make it simple, it's supposed to be activated in cardiac hypertrophy, and it's clearly repressed in heart failure with reduced ejection fraction and notably in DCM. So we had generated such a model of inducible deletion of the gene in the adult heart. And over the years, we have performed a number of omics screen for messenger RNAs, microRNAs, proteins. And altogether, these studies pointed that beyond the expected downregulation of a number of sarcomeric genes, which are really the canonical identified target of SRF, we also had a very profound alteration of genes and protein involved in energy metabolism. And in fact, among the downregulated genes and protein, it was a majority involved in these paths. So we decided, because SRF was involved, activity modulation or changes was involved in different type of cardiomyopathies, to compare our data sets with a number of other deleted cardiomyopathy models data sets available in the GEO database, and trying to look for kind of common pathways, pathogenic pathways which could emerge from this research. And we identified some common pathways involved in energy metabolism, and notably one of them, striking difference common to different models, was a perturbation in NAD coenzyme salvage pathway. And the most striking difference was presumably this activation of this kinase gene called NMRK2 for nicotinamide riboside kinase. And as you can see here, this is our own transcriptomic screen, which is available in the GEO database, where we see that the NMRK2 kinase is involved at early stage, pre-symptomatic, just 8 days after deletion of SRF, and it will rise at 25 days and furthermore at later stages of the disease when the dilated cardiomyopathy is really installed. And when we looked at any other models of dilated cardiomyopathy, we found a similar pattern of induction of IDH2, and here of NMRK2, and here I highlight, for instance, the fact that it's very robustly upregulated in models where you have mutation in this isocitrate dehydrogenase 2 gene, which actually we heard about by Heinrich Steckmeyer in a very nice webinar last month, or not this month actually, just 2 weeks ago. And so in this model too, there is injection of NMRK2, and then a number of different models such as the transferrin receptor knockout, HDEC3 knockout. Here, this lamin A knockout, which is known to trigger Emery-Dreifuss myopathy associated with dilated cardiomyopathy, and other models where, for instance, you have regulators of mitochondrial biogenesis which are deleted. Or here, it's the phospholambda mutation, which is known to create dilated cardiomyopathy in humans. And in the mouse model, it was very clear that this gene was highly upregulated at even early stage prior to the establishment of the dilated cardiomyopathy. So what is this NMRK2 doing? It's a kinase which is using this substrate called the nicotinamide riboside, and I will say NR for now, which is the nicotinamide, the nucleoside form of the vitamin. If you want, there is nicotinamide and the ribose associated. And as a kinase, it simply uses ATP to phosphorylate this ribose, this nucleoside, to generate the NMN, the nicotinamide mononucleotide, which is then, in the process of synthesis for NAD, fused with an ATP moiety by the NMAT enzyme to generate the dinucleotide. And as you know then, as I mentioned previously, NAD is then used as a coenzyme or in its signaling functions. And at the same time, we observed this very strong induction of NMRK2, which, by the way, was a new pathway for NAD synthesis, which was characterized in 2004 by Biegnanovsky and Brenner. There was also a downregulation of this other pathway for NAD synthesis, which I mentioned, I alluded before, which is the NAMPT, which had been discovered just a few years before, actually, and which is this very important enzyme which is able to use NAM to regenerate NMN and then NAD. So one could wonder what is the difference between, for the cells to generate NMN by NMRK2 and NAMPT, because the end result is NAD anyway. So one of the major differences maybe comes from the nature of their substrates, where for the NMRK2, as I said, it's just a nucleoside. It's a single, step to generate the NMN. For the NAMPT, it has to add the full backbone of ribose and phosphate to the nicotinamide moiety to generate the mononucleotide. And this PRPP, the phosphoribose pyrophosphate itself, needs to be generated for the nucleotide synthesis biosynthetic pathways from glucose. And it has a cost in terms of ATP. So if you do a simple stoichiometric calculation, you could say that the NAMPT pathway to generate NMN, the common product of NMRK2 and NAMPT, requires 4 high-energy phosphate bonds, because basically there's 1 ATP here, 1 ATP converted to AMP, so 2 phosphate bonds. And actually, NAMPT, also not a kinase, is using ATP in an autophosphorylation step. So it's altogether these 4 high-energy phosphate bonds, where finally NMRK2 pathway costs only 1 high-energy phosphate bond. And so that's the kind of things that directed us toward looking at whether NMRK2 could actually be an energy stress response gene. And in fact, we showed in vitro that it's responsive to MPK activation and actually through recruitment of PPAR alpha on the promoter of NMRK2. On the other hand, I must say that we still don't know exactly why NMPT's decline is repressed in this model, but interestingly, it's actually a common feature of most heart failure models, and nobody knows exactly how NMPT gene is regulated in that context. So beyond the mice, I just wanted to mention that on top of this shift from NMPT to NMRK2, there are some other genes which are modulated, and notably this anti-nucleotidase, ectodermic nucleotidase, which is also known as CD73, which has been shown to be important for the import of NR when NMN is the external precursor. So it seems like everything is converging in this failing heart to use NR rather than nicotinamide to maintain NAD. Now we wanted to know also if this occurring also in human DCM, because so far I talk only on mice. And we have started to develop a collaboration with Jean-Noël Billot, bioinformaticians at Ejection, to try to build a new pathway that we will make available for the community later on the IPA platform, and to be able to interrogate the variations in gene expressions involved in NAD metabolism in various context disease. And now we decided we have started to apply that at a number of data sets that we found in the GEO database. And for instance, one generated by this consortium, which is called Magnet at UPenn. Here, we selected a subset of patients, Afro-American males over 50 versus non-failing controls as an example, because it's actually a population which is at higher risk of developing heart failure when it develops dilated cardiomyopathy. And as you can see, we find, for instance, in this analysis, a reproduction of the changes that we had observed in mice, mostly an upregulation of NMRK2, sorry, but there was also in that case overexpression of NMRK1, and the repression of this NAMPT enzyme. So this basic pattern of loss of NAMPT and increase of NMRK2 is conserved, as well as this induction of this nucleotidase that I mentioned just before. And as regard the NAD signaling pathway, we found some changes in PARP enzymes going down or up, and CD38 is clearly robustly and systematically downregulated in this context, in these patients. And that is true in other groups beyond Afro-American males, but white Caucasian as well. On the side of the tryptophan de novo metabolism, which can lead to synthesis of NAD through rather a complex and multi-step pathway. There were some changes, but we are not sure, of course, that this is occurring in the cardiomyocytes, because cardiomyocytes are not known to express high level of these enzymes, and that could be a signature of changes in the NAD metabolism of immune cells which are present in the myocardium and could be activated in the context of dilated cardiomyopathy. So that was quite interesting to see the same kind of pattern between mice and humans in that case. Here I'm just showing another example of another NRSX to see if it's just not that one. As you can see, there is this downregulation of NAMPT, and there is this upregulation of NMRK2, which is actually, I must say, less impressive than what we saw in mice. And the reasons for that is maybe that NMRK2, on the contrary to what we saw in mice, has at baseline in a non-failing heart a relatively high level of transcript expression already. It's actually higher in terms of RPK and normalized read in NMRK2 than for NMP2. But so there is still a slight increase, and finally, the end up result is that the ratio of NMRK2 and NMPT is really increased in the context of disease in dilated cardiomyopathy, but actually, as we can see in this data set, as well in ischemic cardiomyopathy. And actually, our published paper 2 years ago in Circulation was in agreement with this more recent analysis that we have performed, because we had observed at the protein level an increase in NMRK2 protein and a decline in NAMPT protein in the human failing heart from patients with dilated cardiomyopathy. And so again, here at the protein level, there is a rise in the NMRK2 to NAMPT ratio in terms of biosynthetic pathways for NAD. So based on this premise, we had started a collaboration, notably with Charles Brenner at the University of Iowa, who was the discoverer, as I said, of this NMRK2 pathway in yeast and human cells as well. And we performed an experiment where we decided to feed our SRF knockout mice with an NR supplemented diet and control mice as well as for control. And we performed a metabolomic targeted analysis with collaboration with Charlie to analyze the level of NAD and some of the intermediates in the NAD synthesis or NAD degradation, such as this deamidated form of NAD, which is known to rise when you stimulate NAD synthesis, or this methylated form of nicotinamide and oxidized form, which are supposed to be kind of waste product of elimination when you have a lot of NAD. And as you can see here in the results, and this has been published already, so I will be brief, but basically what we found is that the NR supplemented diet is able to restore the 25 to 30% decline in myocardial NAD level that we had observed in these mice in the heart, and that were restored to normal by the NR diet. And of course, this is an abundant metabolite, so the changes are maybe more moderate. But when you look at some of these derivatives, which are normally at very low level in the normal diet, both they are in the controls and in the mutants, you can see the impact of the NR treatment on the myocardium is really clear and really rising considerably the production of this metabolite, suggesting that we have indeed with this diet kind of induced again the cycling of turnover of NAD. And on top of that, important question was, of course, the impact on the function, the cardiac function, and very nicely, this NR diet that was able to preserve left ventricular ejection fraction almost to normal. There was still a slight difference with control, but it was a nice preservation, above 60%. And very nicely, it was blocking or limiting the eccentric remodeling, as you can see here from the left ventricular and systolic diameter and left ventricular and diastolic diameter. And there was also a kind of trophic effect on the thickness of the left ventricular wall, which was interesting with this diet. So we had explored this possible mechanism, but it's very complex because, as you have seen, NAD has a pleiotropic functions in the cells. One of the things we were able to show is that while we actually did not see really major changes in the maximal oxidative capacity of mitochondria, in terms of enzyme activity, we saw a nice restoration of the citrate synthase activity, which is reduced in the myocardium of these mice, and which was back to normal with the diet. And there was also a stimulation of this ATP citrate lyase enzyme upon NR feeding, which was also occurring actually in the controls. And altogether, this suggests that NR is kind of stimulating the citrate metabolism, which is taking place, of course, in the mitochondria, but which can also be exported and be used in various processes, such as lipid biosynthesis and notably cardiolipin synthesis, as shown in publication in Cell Metabolism in 2015. And so that could be one of the action of this treatment on stimulation of synthroid metabolism, and maybe like that improving or helping the mitochondrial metabolism. But this has been published, so I would like now, rather to the rest of my talk, to shift to a more new result that we obtained. And we specifically decided to study the role of this nicotinamide riboside kinase NMRK2 in the heart, because there was very little known at the time on the role of this gene in mammals. So we had generated classically a loss of function model by global knockout of NMRK2 gene, and a gain of function model by cardiac-specific overexpression of NMRK2. And I'm going to give you some of the data that we have acquired on these models. So this is the scheme of the inactivation of the wild-type NMRK2 gene. So basically, we use a COM clone, that is an elimination of the whole coding exons of the gene, which is it's a relatively small gene, so that was easy to do, presumably. And that is replaced actually by a LAG-Z. The phenotype of this man has started to be explored by PhD student Cynthia Tanus in my group at the level of the heart, and by Robin Delou in the muscle. What is clear, we use this LAG-Z insertion to actually validate what was already known from transcriptomic assays, but clearly this gene is specific from striated muscles. So we can only find beta-galactosidase activity, which is under the control of the endogenous locus of NMRK2, in the heart and in the skeletal muscle. And there is absolutely no expression in non-muscle cells, in non-muscle organs such as liver, brain, or others. The protein is also expressed in the heart. We cannot detect it in the liver, which is in agreement. Here it's just showing the level at baseline in the wild-type heart. As I told you, at baseline is relatively low. Here it shows the robust induction of the protein in the context of the SRF knockout. The muscle phenotype of these mice, we have published a small studies 2 years ago, is relatively mild. There are some minor changes in the adaptation to endurance exercise training, but nothing really major. So we mostly focus on the cardiac phenotype of these mice, and they happen to have one. Until 2 months of age, we could not see difference in left ventricular ejection fraction or cardiac dimensions. But starting at 5 months and later on with progressive aging from 5 months to 24 months, we followed these mice. We saw some changes in the cardiac in the cardiac function. First, I would like to mention that these mice present a small, slightly smaller body weight compared to controls, which maintains over time with aging. It doesn't affect the cardiac mass ratio, the left ventricular mass index, which is equivalent between the wild type and the cow mice. By the way, the wild type are in green here, the cow in red. But we detected the kind of subclinical left ventricular dysfunctions and dilatation that take place with aging in the mice, notably here at the level of the left ventricle and systolic diameter, which is increased already at 5 months, and the difference trying to amplify with age between the mutant and the controls. To some extent, but less for the LVEDD parameter. And finally, altogether, what it shows is that there is a progressive decline of left ventricular ejection fraction in these mice with age. However, this is never developing to the point to overt heart failure. Clearly, the mice live for 24 months, so you can imagine that already, that they don't really have severe heart failure. And moreover, the cardiac output remains relatively stable. So it's really more a change in the macro structure and some probably the contractility of the heart, but it remains subclinical, I would call it. As regards the expression of genes involved in NAD biosynthesis or signaling, we looked at NMRK2, NMRK1, and AMPT expression over the time. And while NMRK2 expression was declining slowly but remained reasonably good, above 50% at 24 months, for instance, compared to 6 months, there was a more clear decline in the expression of the other isoform, NMRK1, at 24 months, and also of NAMPT. And finally, for the SIRT3 and SIRT1 expression, that was mostly declined at 24 months, but that was kind of identical between the control and the mutant mice. This is end up with a change in NAD that is actually occurring only relatively late in life. So it seems like for the first major part of the life of these knockout mice, they are able to maintain normal cardiac NAD levels, presumably because NAMPT levels at this age are sufficient to maintain this NAD pool. And it's only at late age when the other isoform, NMRK1 and MPT, are down, and that in the mutants you don't have NMRK2 anymore, that eventually we start to see a drop in NAD myocardial levels. But actually, this is coming relatively late, and as you can have noticed from the previous slide, there is already subclinical cardiac dysfunction at earlier ages. And so we were curious to know what, and we started to look a bit at the localization of the protein in the cardiac cells. And we found that it's actually at the baseline when it's not overexpressed, which is just in the wild-type heart, mostly localized at the level of the intercalated disc and along the sarcolemma. And partially overlapping here with the vinculin, which is a cytoskeletal linker between integrins and the actin cytoskeletal inside the cells. And so that reminds us in a way that, in fact, this gene, NMRK2, had originally been cloned under another name, which was muscle integrin binding protein, MIBP, or integrin binding 1, BP3, binding protein 3. And some time ago, and it was shown to actually, so I must say it's actually a partial fragment of NMRK2 with that, like a splice variant, which was cloned by that. And it's been shown to interact with the alpha-7 beta integrin complex, which is linking the actin cytoskeleton to laminin and the collagen extracellular matrix protein. And here it's been shown to interfere with cell adhesion and laminin matrix deposition when MIBP is overexpressed artificially in these myotubes. And more recently in zebrafish, this NRK2B analog, paralog of NRK2 in mammals, was shown when it's in active knockout in these zebrafish embryos to alter the cell adhesion and the morphogenesis of muscles. So there was some indication that there is a role also for this NMRK2 MIBP protein in some structural role in the context of this alpha-7 beta integrin laminin interaction. So we're curious to see if this is happening in the mutant too. When we look at integrin beta-1 and integrin alpha-7, we could not really see difference during the early stages of life. And it's only at about 24 months, and maybe it's occurring a little bit before, we don't know yet, but definitely not at 12 months, that we kind of see a downregulation of integrin alpha-7 in that case, whereas integrin beta-1 remains stable over the time. So this is not, the loss of NMRK2 is not affecting very directly the expression of these integrins. On the other hand, what we could find as an earlier phenotype, and about the time when the phenotype is installing in the mice, is an alteration in the laminin organization in the myocardium of these cow mice. And in fact, what we could see that there is a kind of aberrant pattern of the basal lamina, which is highlighted here by an anti-laminin antibody, which is the partner of this integrin alpha-7 beta-1 integrins complex. As you see in a normal wild-type myocardium, it's a rather thin and well-delineated basal lamina, which is highlighted by the laminin antibody, whereas here it's fuzzy and seems to be wider between the cardiomyocytes. And the electron microscopy analysis actually confirmed this impression, where you have this thin anisotropic basal lamina in the wild-type myocardium, and in many times in between the cardiomyocytes in the cow myocardium, you have enlarged space and kind of amorphous material instead of a real basal lamina. And this is accompanied by collagen fibrase, and I'm not showing it here, but red serous staining showed that there is indeed fibrosis, cardiac fibrosis in this mutant. So that's another additional potential role of NMRK2, that it's, as in the zebrafish or in the myotubes as shown previously in those other models, involved in some extent in a kind of structural role in the establishment of the basal lamina. Finally, I told you that we were developing a gain-of-function model, and I will give you a brief overview of the results, and I finish with that we obtained with these mice. So it's transgenic mice that we obtained by collaboration with Garrett Lavery at the University of Birmingham, who's interested in MRK2 function in skeletal muscle, notably. And he had generated these transgenins which can be induced by removal of stop cassette by pre-excision, and so pre-excision, sorry. And so we breed these transgenic mice from Garrett with transgenic mice overexpressing the recombinase inducible by tamoxifen under the control of the alpha-myosin heavy chain promoter, so in cardiomyocytes only. And so we can obtain overexpression of NMRK2, which is targeted to the cardiac cells. It's a very efficient system, and we had actually a very robust induction of the NMRK2 transgene, maybe a bit even higher than what we would have hoped, because it's very high. But in some disease contexts, it's not hard to see, and for instance, in the SRH knockout, we are sometimes about 50-fold induction of NMRK2. So we are above the physiological, pathological level, but still in the range. There was no impact on the expression of NAMPT, but of the overexpression of NMRK2. But there was definitely, there was an impact on SIRT1 expression, which was mild but significant. So there was a kind of induction of the SIRT1 expression level. And there was an impact when we tested, I haven't explained that, but we looked at this transgenic mice and control mice with normal diet in gray, or with NR supplemented diet, which are black here. And as you can see, this doesn't change much to gene expression level, but for the NAD, we could see that the overexpression of NMRK2 by itself, and in top, in addition with an NR diet, is able to stimulate increase about 25% of the myocardial pool of NAD, which seems to be little, but in the heart, it's actually very hard to increase NAD above the physiological level. So I think it's quite significant already. We followed up the impact of NMRK2 overexpression of several months in these transgenic mice, because after all, it's upregulated gene in disease context, and we wanted to be sure that it doesn't have a pathological impact by itself, like many genes sometimes which are overexpressed in heart. And at least for above 8 months follow-up, with or without NR supplemented diet, we could not see major differences in terms of left ventricular ejection fraction or end systolic diameter. So it doesn't seem to be overtly pathogenic at all in that context. So when we stimulate this NMRK2 NR pathway, again, we produce NAD, and NAD can be used by many different pathways, as I told at the beginning in my introduction. But one of the target pathways that we can imagine would be beneficial to activate is this SIRT1 deacetylase, which is using this NAD to remove, as I said, the acetyl group from proteins and put it on the adiporibose moiety of the NAD. And this enzyme has been involved in a number of critical response for the cells to energy stress and resistance to oxidative stress in a number of different cell types. It's also involved in insulin secretion. It's very clearly important for mitochondrial biogenesis and process important for energy metabolism, such as gluconeogenesis and lipid homeostasis. It has also targets involved in the modulation of apoptosis and insulin sensitivity and tumorigenesis. So in the heart, it's been studied by several groups, a whole knockout of SIRT1, and it was shown known to have a presumably a cardioprotective role. And we have recently re-explored that by generating a more cardiac-specific knockout of SIRT1, so to avoid the potential interference of losing SIRT1 in the different type of organs, to really know what was the function of SIRT1 in the cardiac cells by themselves, using, again, a Krylak system to inactivate the SIRT1 allele. At baseline, I will not describe it here, but it's been published last year. There is a progressive installation of also a cardiac subclinical phenotype at baseline in these mice. But mostly, maybe what is most striking is when you subject them to a stress, such as pressure overload triggered by TAC, a few 2 weeks after triggering the deletion of SIRT1, and follow up for a period of 8 weeks. As you can see, this procedure, which is known to induce cardiac hypertrophy, is inducing cardiac hypertrophy to somewhat a similar level, but a little bit more in the SIRT1 cow TAC compared to the SIRT1 flox mice that we use as control, so they had no Crete engine. And in terms of cardiac function, also this loss of SIRT1 is sensitizing the mice to the stress, and they have a more deeper loss of left ventricular ejection fraction and increase in end systolic volume. So one of the, and actually, sorry, I just finished with that. Also at the terms of mitochondrial respiration, the fact TAC by itself is inducing, an alteration of the mitochondrial oxidative capacities, but it is further increased by SIRT1 depletion upon the context of TAC at the level of the use of palmitoyl-CoA, pyruvate, for instance, or other compounds such as succinate, which stimulate complex 2 respiration. And there is also a decline in citrate synthase activity and to some extent to cyclotron C oxidase. Our group has also been interested in SIRT1 in previous publication, and notably Christophe Lemaire is an investigator in our group who's shown a new function for SIRT1 in the resistance to endoreticulum stress process, which is actually ER stress happens to now discovered as a major pathogenic factor in different forms of heart failure. It's been shown in HFrEF and HFpEF as well. And so it's quite interesting to see how SIRT1 interfere with this ER stress pathway, which, as you know, is linked to alteration in the folding and maturation of proteins synthesized in the endoplasmic reticulum, which are mostly proteins targeted to cell membrane. And what Christophe has shown together with Alexandre Prolat and Julie Pires is that he identified a new role for SIRT1 in targeting this elongation initiation factor 2, which is normally phosphorylated by one branch of the ER stress pathway, and that triggers this activation of this transcription factor ATF4. And when it's overactivated, that process together with the other ER stress branch pathways lead to mitochondrial apoptosis and cardiac dysfunction. And what he showed is that the deacetylation of this EF2 by SIRT1 is kind of lowering or blunting the phosphorylation of EF2 and moderating this response. And at the same time, in a more recent publication, he showed that it's stimulating this other elongation factor 2 in a more indirect way. We don't know exactly the mechanism between these two links, but SIRT1 is required for that, and that promotes autophagy and mitophagy and so cardiac protection. And so since this research was published by Christophe in the group, we decided to quickly look at the NMRK2 TG mice response to a model of ER stress, which is, I recognize immediately, not a pathophysiological model, but we inject tunicamycin. So it's just really to solely focus on ER stress and see quickly if a gene or protein is important for ER stress response. So we inject this tunicamycin by IP, and at the same time we injected nicotinamide riboside by IV and subcutaneous injections for 3 times at the time of injection of tunicamycin, 24 hours and 48 hours, and then we sacrificed the day after. And what we could find is that first, tunicamycin, we had seen that previously, is lowering the NAD level in the myocardium. So you can compare here the blue non-treated control mice and the wild-type treated with tunicamycin. And the NR feeding was actually able to increase the acute injection, especially like that, are robustly increasing the NAD levels in the heart in the wild-type non-treated or even in presence of tunicamycin. And with the transgene NMRK2 overexpression, we have basically the same response, which is over-amplified due to the presence of NMRK2 overexpression. What we found also is that while this tunicamycin stress is triggering the expression of this cardiac stress marker BNP in the myocardium, in the ventricular myocardium, which is blocked by the NR injection, this does not seem to occur in the transgenic mice, which seems already to be less responsive to tunicamycin in that regard. When we look at the fractional shortening in these mice, we saw that tunicamycin, as you can see, is inducing an acute reduction in fractional shortening. But NR treatment in the wild-type mice, which have normal NMRK2 level, doesn't really rescue this aspect of cardiac contractility. Whereas in the presence of the NMRK2 transgene, there was a reduction by tunicamycin, but in presence of NR diet with the transgenic mice, it was able to preserve the fractional shortening, which is quite interesting. When we looked at the mitochondrial oxidative capacities, what we found is that this tunicamycin injection is actually affecting the capacities of the mitochondria to use pyruvate at the level of complex 1. We use the permeabilized cardiac fibers protocol for that. And it's also altered, so not significantly, the close to significant but not yet when you have both complex 1 and complex 2 activity, which is used. So presumably the main difference is at the level of complex 1. And also when you stimulate global maximal respiration, if you want, by stimulating the complex 4, you see that the decline with tunicamycin of the oxidative capacity. And this was actually nicely preserved by NR treatment in the wild-type context, even in absence of NMRK2 for all the parameters. Again, the NMRK2 transgenic mice, even without NR, seem to be less affected than their wild-type counterpart in presence of tunicamycin. And so in a way, they didn't need so much the NR addition in the diet. So we looked a bit also at the different markers of ER stress response in that context. And I'm showing you what we have now. We are continuing this analysis, but basically, again, like for the other parameters I showed you previously, mostly we saw a robust induction of these parameters upon tunicamycin markers of ER stress as expected upon tunicamycin injection in the wild-type mice, such as here the CHOP mRNA. Here, sorry, I can't see this part of the screen because you have the ITF6 mRNA, and which is also a marker of this branch of ER stress, or here this splice form of XBP1 mRNA, which is also a transcription factor involved in the regulation of chaperone and apoptosis. And of these chaperones, which are located in the ER itself, like GRP78 and GRP94. All of them, strongly or robustly induced by tunicamycin in the wild type, bring back to normal by NR treatment. But in the transgenic mice, even before not having NR but subjected to tunicamycin, there is less response in that regard. So there seems to be a cardioprotection against TDR stress activator provided by the overexpression of NMRK2. So now, for some reason, I lose the ability to go to the next slide. Should have not moved this. That's something really strange. Just moving the pictures is like...
Matthias, we went quite over time. Can you find a way to wrap up quickly so we have some time for questions?
You're muted now, Matthias. You're actually muted, so oh, there we go.
Okay. I have to, for some reason, my system is frozen, so...
Okay. Let me try and stop sharing a screen.
Okay.
There we go. Okay.
Are you seeing better now?
Yes. Well, you are not now on full screen, but that's fine. Depending on how long you've got left.
I'm done. I'm ready. You see, that's my thanking slide, so I'm done. So I would like to conclude, and I will not put it full screen. I think it's easy. You can read it, right? To say that what we have seen, basically, to summarize our results, is that we have seen changes in AMPT2 and NMRK2 pathway for NAD synthesis, and we think that they find a new type of metabolic shift in the DCM failing heart in mice and in human patients, as I showed you. What we have shown is that at baseline NMRK2 is, I think, a palliative salvage pathway that is maybe not required for cardiac function and maintenance of NAD levels before mid to late life stages in mice, but it has an alternative function in integrin-laminin interaction, obviously, at slightly earlier ages. Finally, NMRK2 overexpression, as I showed you, is not pathological per se, so it's not because it's overexpressed in disease that it's a pathological mechanism, and rather we think that it has the potential to be a cardioprotective mechanism. So obviously we need to develop that research further and test more pathological models. And finally, nicotinamide riboside treatment emerged as a new therapeutic avenue for metabolic therapy of heart failure, and I will finish with that by reminding that Rong, at the time we had this paper out, wrote an editorial and asked a question of whether rising NAD in heart failure, was it time to translate? And I think now in 2020, we have the response to that since the first phase 1 trial published in 2016 by Charles Brenner. There has been a number of studies which show that first NR is safe to use in humans. It's efficient to rise NAD levels in the blood, and that was actually shown also by Rong Chan in a publication. And it's been over 46 clinical trials which have been started over this period of time in various contexts: aging, metabolic syndrome, cognitive troubles, and very recently, Rong and Kevin Aubrey in Seattle have started a systolic heart failure clinical trial using, trying to see if NR could be useful in that context. And I think we are all looking forward to the result of this clinical trial in the near future. On our side in France, we are trying to set up also a clinical trial and not really focusing probably more on idiopathic dilated cardiomyopathy patients. And I'll finish with that. Would like to thank all the collaborators, but I already mentioned them in the time course of my talk. I apologize for the long time and the bugs. I thank the team member and our support, and I'm welcoming questions. Sorry again. So should I stop the screen sharing, or you did?
Great.
We can't hear you.
Thank you, Matthias. That's a fantastic talk, a lot of information. We have already quite a number of questions lined up here. What we're going to do is I'm going to look at these questions and either to read out the question or paraphrase it, and then Matthias will answer them. So I apologize in advance if I said somebody's name wrong. I will try my best. And also because of the time restraint, we will make the question short and the answer short as well, so we can cover as many as possible. And if we didn't get to answer your question, please contact Mathias separately, and I'm sure he will be happy to explain more and have a further discussion. So, okay, so the first question has come from Luc Bertrand. Hi, Luc. He says, excellent talk, Mathias. There are now novel specific direct and potent AMPK activators. Would that be useful for basically enhanced NAD metabolism pathway?
Yes. Hi, Luc. So yeah, it's a very important question. I think clearly what we had shown as regard the link between AMPK and NMRK2 is the fact that it's in vitro so far. But clearly in cardiac cells, if you put AMPK activators or if you put cells into glucose deprivation, which is known to activate AMPK, there is a clear, robust induction of NMRK2 gene. But really now, I think it's an important question that Luc is asking. We should consider whether using this novel type of AMPK activator is activating NMRK2 in vivo in the heart. That's the next question, but we have not done that yet.
Great. So the next question is from Heinrich Peckmeyer. Hi, Heinrich. So it says, beautiful, Mathias. Heinrich is wondering what are the consequences of increased ACL activity?
Yes, well, ACL activity ACL is an enzyme, as I said, which converts citrate, which coming from the Krebs cycle. That's Professor Peckmeyer beautifully explained to us a few weeks ago. And that goes to the cytosol, and then it's converted to acetyl-CoA. And this enzyme has been shown, at least in muscle, to be very important for the lipids biosynthesis, and notably the lipids. And it was shown in that study, that it was then improving the mitochondrial function. So that was in skeletal muscle. We don't know in the heart. If we have not gone to this step, we have to perform, for instance, a cardiolipid profiling, as we have done for other models in the past in our lab. So we have the expertise for that, and we'll see. That would be maybe a potential impact of this stimulation of ACL activity, because cardiolipin profiles are usually altered in heart failure, and that one consequence of the NR treatment stimulating ACL could be, but I'm speculating, normalization of the cardiolipin profile.
Great. So the next question is from an anonymous attendee. It is about NRMK2 in the SRF knockout heart, and it is asking whether the induction of NMRK2 in the SRF knockout suggests that SRF is normally a negative regulator of this gene. And so in this knockout, the NMRK2 transcription, is it depressed?
Right, yeah, that's an interesting question. Yeah, that was the first idea, I must say. But then we had actually cloned the NMRK2 promoter, and I suggest to refer to this publication in Circulation that we had in 2018, where there is a figure on that. But just to briefly summarize, in fact, no, there is a SRF binding site in the promoter, but when we mutate it, at least in classical experiment with luciferase reporter, when we mutate and block the binding of SRF to this promoter, we have a decline of NMRK2. So strangely enough, it seems to be a positive regulator of NMRK2, yet when SRF is inactivated, we have induction of the NMRK2 expression. And that, we believe, is coming from other activating pathways which are raised upon SRF deletion and upon the establishment of the heart failure. And that could be very well this AMPK signaling that Luc Bertrand mentioned before. And that would overrule the loss of SRF. So what we saw in vitro is a loss of about 50% of basal expression of NMRK2 when we just remove the card box, which is a binding site for SRF. But the PPAR sites are independent from these SRF binding sites, and presumably they can replace the loss of SRF and stimulate strongly the gene. I hope this answers the question.
Okay, so the next question is also from an anonymous attendee. it asks whether there's any changes on SIRT6 in the on those knockouts.
Did not came out from our transcriptomic screens and neither from our proteomic screens. I don't know if you would have seen that, but at the transcript level, we don't see any change. That's all I can say for now.
Okay, and the next question comes from Gabriel Chiatarita. It says, great talk, Matthias. And wondering how does it look for the total mitochondrial protein acetylation in the knockout or overexpression mice?
Hi, Gabriel. Nice to hear about you, Vay. Yeah, very pertinent question. And actually, I kind of stay aside of this debate in this talk, but yeah, it's a very important question. In the NMRK2 knockout mice, we haven't tried that. What we can say is that normally, I would refer briefly, it's not really the question, but in the SRF knockout, for instance, when we treated them with NR, what we see is, and there is overexpression of NMRK2, we saw actually an increase in the acetylation of total protein, but we have not looked at them specifically at the mitochondrial pool. And for the NMRK2 cow mice, we were doing that, but I don't have the answer yet.
Okay, so the next one comes from Sima Hadipur-Lekmesari. The question, well, at first, a great talk, Matthias. There's two questions. One is that, is there a similar variation in the level of MRK2 between human DCMG mutations compared to various mouse genetic models? The second one is whether the knockout is tachycardic. Do you see any increase in arrhythmias?
Right. So to respond to the first questions, that's what I showed at the beginning of my talk. It's basically there is also an increase of NMRK2 transcription in the human DCM patients. I cannot pinpoint a mutation because this is data sets which are incorporating patients with idiopathic dilated cardiomyopathy. So it's not specifically one gene mutation. But so there is a parallel. There is also an increase of NMRK2. It's just the fold change is maybe less important than it is in the mice. And for the second question, we haven't looked at that in the knockout mice yet. We have started to look at it in the transgenic mice overexpressing NMRK2. We don't see evidence of it, at least. No sudden death, for instance, sudden deaths, which would have lead to a drop in the population of the NMRK2 knockout over the time. So they live relatively well until 2 years long. So if there was significant arrhythmia and risk of sudden deaths, I would say we should have seen it. We haven't seen that.
Matthias, sorry if I don't mind if I'm interjecting. Now I remember why I know you through Gareth Lavery. And we actually did phenotype some of the NRMK2 overexpressors. And after this webinar, I will share with you some of the information.
Right. Good.
Now it's clicked.
Yes, you were involved in that. That's what I was telling you. I was thinking
Sorry, this was a discussion before the webinar.
Right. We had a Skype meeting. We didn't.
So yes, so now it's clicked and I can show you some data.
Right.
Great. Okay. And now you find a friend in the club.
Right.
Yes.
Okay. So the next question is from Emma Robinson. Emma says, thanks very much for your comprehensive talk. She's interested in change in PARP enzymes. The PARP is, besides being involved in DNA repair, it could also influence DNA methylation. So she was wondering whether there's any changes in the components of, let's see, in the heart, whether the DNA methylation pattern in the heart are changed in the DCMs in humans and animal models. Has anyone done systemic knockout out of individual change components of PARPs to see how these changes in cardiac gene expression are due to change expression and activity of PARPs alone compared to what is the effect of the disease?
Right. This is a rather long and complex question, and a little bit beyond my own scope of expertise. So I prefer not really to go into detailed response for that. I know there has been knockouts of PARP1, for instance, and some of them have been published. I'm not quite sure that they have looked at DNA methylation in this model, so it's certainly an interesting idea to do, but I have no clue. I'm sorry.
Great. So maybe Emma, you can contact Matthias and give him some new ideas.
Yes, exactly.
So the next question comes from an anonymous attendee. Could you please repeat the most direct protein substrate of NRMK2 kinase? Any specific pattern on the phosphorosides of those substrate proteins, like to specify protein families, linked to specific protein families?
Right. Well, no, actually, maybe I was not clear enough, but I don't think we know that this kinase can have a protein substrate. It's really a kinase which has an affinity for nucleotides, especially nucleosides, for instance, especially first for this nicotinamide riboside. In the original publication by Charles Brenner, he showed that eventually there could be some other nucleosides that can be phosphorylated by this kinase, but really it's a kinase phosphorylating a nucleoside, and so far, at least, we don't know any protein target for this kinase.
Okay, the next question comes from Lucy Carrier. Exciting talk, Matthias. Congrats. Looks like the NMRK is expressed in human iPS cell-derived cardiomyocytes. Have you had a chance to perform a CRISPR-mediated knockout?
Hi, Lucy. Nice to hear from you. I see you try to get some insider information here. No, but yeah, we have tried. We have started to do actually a collaboration with Jean-Sébastien Hulot. Some experiments looking at NMPT first, but also we plan to do it on NMRK2 in iPS. But yes, NMRK2 is expressed in the iPS cells, derived cardiomyocytes. So it's an interesting thing to do. We want to do it.
Great. So the next question from Duha Akintievich. Beautiful talk. So DCM is a hallmark of cardiac inflammation. Other changes in NRK2 transgenic heart in terms of immune cell recruitment.
We haven't looked at that specifically, so I cannot answer. It could be, I don't know. We overexpress NMRK2 solely in the cardiomyocyte cells, but who knows indirectly what it could do to the immune compartment. That's something we could probably look at, maybe in a pathological context, like if we induce DCM or myocardial infarction. Yeah, but no answer yet.
So next question is from anonymous attendee again. Is there an H3K27 acetylation changes in any of your transgenic or knockout animals?
Yeah, as a target of sort one could be, but we haven't looked at it. Yeah, sorry.
Okay, another question from Emma. Are there changes in absolute number of cardiac myocytes in the NMRK knockout mice, even at a young age? I think she is thinking about whether the proliferation capacity has changed.
Absolute number of cardiomyocytes. We haven't looked at that. What I can say is that when we really look carefully at the organization of the matrix, extracellular matrix, as I showed some pictures, there is no obvious major change in the thickness of the myocardium and the number of cell layers that you can see usually in a left ventricular wall. So that's all I can say. But absolute number is, yeah. Some people can do that, I know, but it's complicated.
All right. So next question asks whether the XBP1 splice in the NRK2 transgenic mice is also commented that doing the IV injection it's an interesting approach, and most of the time it's used in vitro, the tonica.
Right. We detected in the NMRK2 transgenic mice did you splice as a form, but it's less than in the wild type because no, sorry, stop the screen sharing, of course, but yeah, no, too complicated to start that over. So it's just less induced in the NMRK2 transgenic mice than it is in the wild type mice. So there is an impact of NMRK2 on this step of splicing. But is it very specifically on this branch of ER stress, or is it more a global effect? Because we saw like most of the markers of ER stress, even the other branches of ER stress pathway, were kind of less activated in presence of NMRK2. That's what I can say.
So this, it seems like this attendee also followed up with a comment and said that they've done some in vitro studies and also looked at mouse models, and so there's a reference cited there. Maybe it's worthwhile to check it out.
Yes, we that's what actually we did. Yeah, we used it in vivo, and that model has been set up by Christophe Lemaire in the team, and yeah, it's a nice. I think that's what this person alluded. So we didn't use it only in vitro. We use it in vivo, and it's good to study ER stress in vivo with this method of injection, and it's published, so we can give the protocol, no problem.
Okay, so here's the last question from also anonymous attendee. Is the structural function of the NMRK2 independent of its catalytic activity? And there's a zebrafish paper from Goody, and they speculated about a post-translational modification of the ECM, such as ADP ribosylation.
Right. I see that. So someone who knows well the literature. Yeah, it's true. Yeah, that in the Goody paper, they suggest that in the discussion, they hypothesize that because they show what is interesting in their study, very interesting, is that they show that dipping, this is a zebrafish, so they just put it in a water bath with NAD. That's easy to do. It's cool. And apparently that is sufficient in a way to rescue the phenotype of the lack of NMRK2. So suggesting that this morpho-structural role of NMRK2 and the laminin and on the ECM requires its activity's possibility to synthesize NAD. And that would then come from ADP ribosylation, maybe of some ECM proteins, and notably there is one which has been described for integrin alpha 7. But this is really, we have just started to attempt to look at that, but it's quite complicated post-translational modification because there is no really antibodies available to really look at that. So you need proteomics, and it's membrane-linked protein, so it's complicated. But that's one possibility, is that there's a localized role for NMRK2 to providing NAD to some ADP ribosylase. For instance, there are ART1, for instance, that could ADP ribosylate some proteins involved in the connection between the cytoskeleton and the membrane. So that's one possibility indeed. Yeah.
Interesting. So that comes to the end of the Q&A. Again, I want to thank Mathias for a very stimulating talk, and thank everybody to stick around for the discussion, and also, of course, to Deborah for putting together the series. All right, we will see you tomorrow again.
Okay, thank you.
Have a good day and good night.
Yes, and sorry for the many bugs again. I'm very sorry. Bye-bye. Have a good day.
Yes, on my behalf as well. Thank you. Really, really great talk.
Thanks.
See you all tomorrow.
Bye-bye.